Good morning, students. Let's continue with the chapter number 3. Next topic, angiosperms. They are different from gymnosperms, where ovules were naked, but in angiosperms or flowering plants, the pollen grains and the ovules are developed in the specialized structures called flowers. In angiosperms, the seeds are enclosed in a fruit. The angiosperm are an exceptionally large group of plants occurring in wide range of habitats. They range in size from smallest Wolffia to tall trees of Eucalyptus whose height is about over 100 meters. सबसे छोटा वॉल्फिया और लंबे ट्रीज में यूकेलिप्टस जिनकी हाइट 100 मीटर से भी ज़्यादा हो सकती है. They provide us with food, fodder. Fodder is animal food, fuel, medicines and several other commercially important products like paper, gum, rubber, resins, sandalwood, etc. They are divided into two classes, dicotyledons and monocotyledons. These are the types of plants on the basis of the type of seed they possess. The dicotyledons are the seeds which have two cotyledons, while the monocotyledons have only one cotyledon. Here we can see in this diagram that monocot plants have the seed with one cotyledon only, while dicots are the plants those have seeds which contain two cotyledons. Examples are given. Grasses are monocots while beans are dicots. Along with the difference in cotyledon number, they also differ in the type of venation in the leaves. Venation मतलब leaves में जो veins होती हैं उनका pattern. तो pattern of veins is known as venation. So dicots have reticulate venation in which the veins appear to be like a network, and the flowers are tetramerous or pentamerous. Tetra means 4, penta means 5. So the flower have 4 members in each whorl. While monocots have parallel venation, as we can see here, the dicot is having a network-like structure, while monocot is having parallel lines for the veins. Monocots are trimerous generally, means they have 3 members in each floral whorl. In the flowers, the male sex organ is stamen. Each stamen consists of slender filament with an anther at the tip. Within the anther, pollen mother cell divide by meiosis to produce microspores which matures into pollen grains. The female sex organ in a flower is pistil. It consists of a swollen ovary at its base, a long slender style and a sticky receptacle known as stigma. Here in the flower we can see the anther. The position of the anther at the tip of a filament, green color filament. This whole structure, anther plus filament, is known as stamen. While in the pistil we can see that at its base it is swollen, forming a structure called ovary, inside which ovules are present, which are converted into seeds after fertilization. A long tube-like structure is seen here that is known as style, and the tip of the carpel or pistil is known as stigma, which is sticky in nature. Let us see the function of these organs. In a flower, stamen is the male reproductive part, while pistil is the female reproductive part. We know that male reproductive part, stamen, will produce male gametes, and pistil, female reproductive part, will produce female gametes. The gametes will fuse and form the zygote. At that time, the ovules are converted into seeds and ovary into fruit. Inside the ovary, we have seen the ovules. Generally, each ovule has a megaspore mother cell. जैसा हमने एंथर में देखा था कि पोलन मदर सेल था, यहाँ पर मेगास्पोर मदर सेल है, वहाँ पर भी मेयोसिस हो रहा था, यहाँ पर भी मेयोसिस होगा। वहाँ पर 4 हैप्लॉइड माइक्रोस्पोर्स बन रहे थे और यहाँ पर 4 हैप्लॉइड मेगास्पोर्स बनेंगे। वो माइक्रोस्पोर पोलन ग्रेन बना रहे थे, यहाँ पर मेगास्पोर में से एक डिवाइड होकर एम्ब्रियो सैक बनाएगा, जो हमें यहाँ पर दिखाई दे रहा है। Let us see in the diagram. The open end is the micropylar end. 3 megaspores degenerate and only one gets mature and divides mitotically to form 8 cells. Out of these 8 cells, 3 of them organize to the micropylar end, 3 of them to the opposite chalazal end, and 2 in between in the center. The micropylar 3 cells are 2 synergids and 1 egg cell, while at the chalazal end, all the 3 are antipodals. In between, the 2 cells fuse their cytoplasm but not the nuclei. Hence, the cell is central cell with 2 polar nuclei. Only this is the diploid structure. Rest 6 cells, egg cell, synergids and antipodals, are haploid. Pollen grains, after dispersal from the anther, are carried by wind or various other agencies to the stigma of the pistil. This is named as pollination. पोलन ग्रेन का स्टेमेन से स्टिग्मा पर पहुंचना is known as pollination. It can be done by the agents like wind, insects, human, animals, birds, etc. The pollen grains germinate on the stigma and forms pollen tube which grow through the tissues of the stigma and through the style and finally reaches the ovule. The pollen tubes enter the ovule from the micropylar end and discharges 2 male gametes. One of the male gamete fuse with the egg cell. The process is known as syngamy and zygote is formed. The other male gamete fuses with the secondary nucleus central cell, which is diploid, and produces a triploid pen, that is primary endosperm nucleus. Because of the occurrence of 2 fusions, syngamy as well as triple fusion, this event is known as double fertilization. Here we can see how the flowering plants reproduce. In a flower, there are stamen and pistil. They produce the male and female gametes. From the stamen, pollen grains are produced, which land on the stigma, which is a female reproductive organ. There, they germinate and pollen tubes are formed. The pollen tubes reaches the ovule. Once they reaches the ovule, they discharge the gametes and double fertilization occurs. It is the unique event to angiosperms only. The zygote develops into an embryo, maybe with the 1 cotyledon or the 2 cotyledons. The pen develops into endosperm, which provides nourishment to the developing embryo. The synergids and the antipodals degenerate after fertilization. During these events, the ovule develops into seeds and the ovary matures into fruit. Let us see the life cycle of a typical angiospermic plant. A plant is considered to be sporophyte and the sporophytic generation. Ovary consists of ovule which produces egg cell, while anther consists of pollen grains which produce male gametophyte, and the gametes are discharged into the embryo sac for fertilization. After fertilization, zygote is formed. The zygote starts developing into the embryo and the embryo converts into sporophyte. So, till the ovule and megasporangia, the generation is sporophytic, but the microspore and megaspores represent the gametophytic generation. Plant life cycles and alteration of generation. In plants, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies, that is haploid and diploid. The haploid plant body produces gamete by mitosis. This plant represents gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophytic plant body. The haploid spores are produced by this sporophytic by meiosis. These in turn divide by mitosis to form haploid gametophytes of the plant. Once again, haploid plant body is obtained. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of generation between the gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals represent them in a following different patterns. There are total 3 patterns. First is haplontic life cycle. In this life cycle, sporophytic generation is represented only by one-celled zygote. There are no free-living sporophytes. Meiosis in the zygote results in the formation of haploid spores. These spores divide mitotically and form gametophyte. The dominant photosynthetic phase in such plant is a free-living gametophyte. This kind of cycle is termed as haplontic. Many algae such as Volvox, Spirogyra and some species of Chlamydomonas exhibit this type of life cycle. We can see here in the pie chart also that the maximum portion of their life is in the haplontic stage, gametophytic stage, which is haploid, while the sporophytic stage is only zygote, that is diploid. The next type of life cycle is diplontic life cycle. It is the type wherein the diploid sporophyte is dominant, photosynthetic and independent phase of life. The gametophytic phase is represented by the single to few celled haploid gametophytes. This kind of life cycle is termed as diplontic. An alga fucus represents this pattern. In addition, all seeds bearing plants, that is gymnosperm and angiosperm, follow this pattern, with some variations wherein some gymnosperms show multicellular gametophytes. Here in the cycle, we can see that sporophyte diploid condition is the major condition of the life cycle, whereas the gametophytic is a very short-lived stage in such plants. The third type is seen in bryophytes and pteridophytes, that is an intermediate condition, haplodiplontic. Both the phases are multicellular and however they differ in their dominant phases. A dominant independent photosynthetic thalloid or erect phase is represented by haploid gametophyte and alternates with short-lived multicellular sporophyte totally or partially dependent on the gametophyte for its anchorage and nutrition is seen in all bryophytes, while in pteridophytes the diploid sporophyte is represented by dominant independent photosynthetic vascular plant body which alternates with multicellular saprophytic autotrophic independent but short-lived haploid gametophyte. This is known as haplodiplontic life cycle, and this is, as I said, is shown by pteridophytes. Here we can see in the diagram also that there may be a short-lived sporophyte in some condition and dominant gametophyte, while in others dominant sporophyte and short-lived gametophyte. So this represents that both the stages are of equal importance and independent survival. Hence, they are known as haplodiplontic life cycles. Interestingly, while most algal genera are haplontic, some of them such as Ectocarpus, Polysiphonia, Kelps are haplodiplontic. Fucus is an alga which is diplontic. So these are some exceptions of the algae which we must remember very well. This is all about chapter 3. We have read about algae, bryophyte, pteridophyte, gymnosperm, angiosperm, plant life cycles and alternation of generations found in all the plants. So with this the chapter is over. If you have any doubts, you are most welcome.